Hello everyone, welcome to the new session of Polyploidy. So we are going to study the polyploidy in the two parts. So this is the first part. So polyploidy was first discovered in 1907 and was thought to be responsible for increasing the number of genomic copies that are heritable. So in this phenomena, that is the polyploidy, what happened? Increase the number of genomic copies. So suppose an organism is having 24 chromosome number, it means 12 pairs. So whole this 12 pairs get duplicated, that is 24 pairs. That means 48 chromosomes. Right? So it doubles the chromosome number. So earlier 12 pairs were present, but now 24 pairs were present. And if we just count the number, then it will be 48 chromosome instead of 24. So this polyploidy is the condition of having more than two sets of chromosomes and also acts as an important mechanism for the plant evolution. So generally the plant is diploid, that is 2n. So this was the 2n, 12 pairs. It is already diploid, right? But this get doubled means tetraploid, that is 4n, when it becomes 24 pairs, right? So polyploidy is the condition of having more than two sets of chromosomes and also acts an important mechanism for the plant evolution. So this polyploidy gives rise to different types of plants, okay, in the due course of time. That is called as plant evolution. Among almost 70% of the flowering plants, that is the angiosperm, have undergone polyploidy several times during their evolution. So now we can understand how this polyploidy is important, because today we are having so many angiospermic plant species, and out of that 70% has been formed because of this polyploidy. Okay, so polyploidy leads to change in the gene dosage. Now what is this? Gene dosage means every gene is present in a certain dose, that is called as doses compensation, right? So if we are having, suppose a plant is having 12 pairs of chromosome, it means that the gene is present in the paired form, that is 2n, that means 2c, okay? And when it get doubles, means tetraploid, then it will become 24, and every gene will present in 4 times. Okay, so the change in the gene dose that causes chromosomal rearrangements, epigenetic remodeling, and reunion of divergent gene regularity hierarchies. So while the cell is going under the polyploidy or the polyploidy, when there is a change in the gene number, okay, what happened? There is a phenomena that is called as chromosomal rearrangement, okay, epigenetic remodeling and reunion of divergent gene regularity hierarchies. So chromosomal rearrangement, because earlier it is having just 12 pairs of chromosome, but now it is having 24 pairs of chromosomes. So the chromosome has to arrange in the nucleus accordingly, right? So these changes may bring out the novel variations or may result in the speciation and reproductive isolation. Now, this particular plant which undergoes the polyploidy will be very different from the parent one. Okay, so there will be a variation in the plant. New character will be visualized and may result in speciation later on. So these are the very important aspects of the polyploidy, and that give rise to the evolution in plants. So polyploids may develop naturally also and by the cytological mechanism like production of unreduced gametes. Generally, the gametes are found by the reduction of the chromosome number. So suppose the plant is having 12 pairs of the chromosomes, right? So the gametes will have 6 chromosomes, half of it, right? So here the chromosome got reduced. But sometime for the polyploidy, in polyploidy, there is no reduction division at the time of formation of gametes. So the production of unreduced gametes are there, or may also occur due to the unfavorable environmental condition. So these are some of the mechanism that undergo in the cell. That is the mechanism like production of unreduced gamete, and sometimes some environmental factor are there that do not allow the cell to undergo the reduction division. And the gametes are having the same chromosome number as the parent cell. So the plant breeders select the polyploids plant in order to improve their agronomic traits. Okay, so plant breeders select those plants which are having the polyploid. Okay, and that will helps in their agronomic traits, to identify the agronomic traits, and some of which were developed by the autopolyploidization. That is a type of polyploidy within the species. Okay, that is the within the species, or allopolyploidization, that is occur in the different species or interspecific hybrids. Okay. Compared to the allopolyploids that are more common in nature, autopolyploids pose a great advantage as these organisms maximize the number of genes and allele variant. Okay, so here allopolyploids that are more common in nature, this is more common, and autopolyploids are less common. Okay, but that is having the great advantage as these organisms can maximize the number of genes and allele variants in the same species, that is autopolyploids. Okay, but here in allopolyploids, 2 different species are involved and the chromosome number get doubled or 4 times. Okay, so some traits like drought tolerance, pest resistance, blooming time, size of organ, apomixis, and biomass increase the chance of polyploid plants being selected for the agriculture. So these are some of the traits that are identified in the polyploids. Okay, these polyploids generally have these types of traits like drought tolerance, okay, pest resistance, blooming time, the flower may get flower early, okay, size of organ, apomixis, and biomass increase. So polyploid have so many advantages. Polyploid increase the genetic base through the development of breeding lines in short time span and restoring the hybrid fertility in the interspecific or interploidy crosses. So in the short span of time, short duration of time, it can restore the hybrid fertility. The plant is hybrid and it is having fertility also, that it can restore the hybrid fertility in the interspecific. 2 different species are involved, that is why it is called as interspecific and interploidy crosses. There are three noticeable evident advantage of the polyploidy comprising the heterosis, that is the hybrid vigor, and genetic redundancy and asexual reproduction. So sometimes in genetic redundancy means there are genes are present in the paired form, okay, and sometimes one of the gene of the pair is do not work normally or it do not express. So on the contrary of that, the second gene of the pair will express that phenotype effect, will express the trait, the character that is responsible. The gene is responsible. Okay. Gene redundancy also causes the burring effect in which extra copies of the wild type alleles conceive the effect of the deleterious allele. So sometimes there are deleterious allele. Okay. So, and sometimes they are also having the wild type of allele for that particular gene. So this wild type of allele do what? It causes the burning effect, because this wild type is having the extra copies and it can mask the effect of the deleterious effect of certain different allele. Okay, deleterious allele. So it can mask that effect. In this way, extra copies of genes provide a protection against inbreeding depression. So this is called inbreeding depression because some deleterious allele causes the depression, means it showing the deleterious effect. Okay, so that is why I call it depression, inbreeding depression. But when the genes are having some extra copies and that gene is having the wild type of allele, it provide protection to the plant as it do not allow the allele which shows the deleterious effect. Okay, so and they are present on the single locus deleterious mutation effect. So in this way, extra copies of gene provide protection against inbreeding depression and the single locus deleterious mutation effect. So why some of the genes show the deleterious effect? Because of the mutation. So if for a particular gene, suppose this is a gene and we are having number of suppose this is A, this is A dash, this is A dash dash, and this is A dash 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 3 times. Now this one, suppose A dash is showing the deleterious effect, okay, due to mutation. There is a mutation in this particular gene. Okay, in fact, allele, these are all showing the one gene, that is A. Okay, and we are having the different set of chromosome having the same allele of one particular gene. So that shows because of the mutation. Now this particular gene A dash dash and dash will show or will mask the effect of this deleterious allele because it is a wild type. Okay, so in this way, sometimes the polyploidy helps plants and provide protection against the inbreeding depression. Types of polyploidy. So there are three main types of polyploidy: allopolyploids, autopolyploids, and autoallopolyploids. So in this session, we are going to discuss about the allopolyploids. Euploid constitute the majority of polyploids. Euploids are the polyploids with multiples of a complete set of chromosome species to a species. So multiples time a complete set of chromosome is present, then it is euploids. Depending upon the composition of the genome, euploids can be further classified as autopolyploids and allopolyploids, right? So tetraploidy is the most common class of eupolyploids. Now, we will discuss allopolyploids. So allopolyploids are also called as alloploids, and they are the combination of the genome from the different species. So we are having two different species, suppose A and B, okay, and from this particular species we will have some set of chromosome, okay. Suppose PP, it is having PPQQ, okay, and we will have PP, and this one is having RR and SS, so we will have SS, okay. So PP and SS we will have in this particular new species. So in this way, there is a combination of genome of the different species. So this is having, this allopolyploid is having the combination of A species and B species. A species is from A species, it is having the P, and from the B, it is having the S types of alleles. So they result from the hybridization of the two or more genomes, followed by the chromosome doubling or by the fusion of unreduced gametes between the species. So hybridization technique is one of the important tool for the formation of allopolyploids. So this process is a key in the process of speciation for angiosperms and ferns and occur often in nature. Economically important natural allopolyploids crops include strawberry, wheat, oat, upland cotton, oilseed rape, blueberry and mustard. So many allopolyploids example we are having. Okay, so strawberry, wheat, oat, upland cotton, oilseed rape, blueberry and mustard are some of the examples because they are economically important and having some or the other importance from the conventional type of crop. Okay, so this is very important. So to differentiate between the sources of genomes in alloploids, each genome is designated by a different letter. For example, the origin of the cultivated mustards have been well explained by the Nagaru in the triangle of U, which with each species represented by the distinct letter. Now, it has been used some genus such as Cucumis to elucidate the molecular mechanism involved in the diploidization tendency of polyploids to act as a diploids. In this study, the allotetraploids was induced by the hybridization process between the Cucumis sativus and Cucumis hystrix, followed by the chromosome doubling. So we will see one of the examples. Here we are showing the same thing with one of the example. Brassica nigra. Brassica nigra is having N8 and it is having BB set of chromosome, and Brassica oleracea from Brassica napus, it is having N19, that is A and CC. So from this, it is having CC. Brassica oleracea is having CC and it is having N is equal to 9. Whereas the Brassica rapa, it is having AA set of chromosome. Now, from here, the CC set of chromosome is taken from the Brassica oleracea and BB from the Brassica nigra, and these two chromosomes are combined to form Brassica carinata, a new species, Brassica carinata. In the same way, Brassica juncea is formed from Brassica nigra and Brassica rapa. So now here you can see and understand how the new species can be formed. One more example. Let us understand how these allopolyploids are formed. Hybridization is the merger of the two genomes within one nucleus, is an important mechanism for creating genetic diversity. So here we are having diploid species 1. So here you can see 3 sets of chromosomes, and diploid species 2. And it is also having different 3 sets of chromosomes. Okay. Now, when they both are merged together, diploid hybrid. This is called diploid hybrid. So, we can have this particular 1 chromosome, and from this we are having 1 chromosome like this. And whole genome diploidization. Whole genome diploidization occur, WGD, and you can see here there is tetraploids, that is allopolyploids. Okay, so genome merger allows for the novel genetic interactions and the allele combinations, which in turn can lead to new phenotypes for the selection to act upon. So genome merger, we are merging the genomes from the diploid species 1 and from the diploid species 2 in one nucleus by the process of hybridization. So this potential for the phenotypic novelty is enhanced in the allopolyploids. Allopolyploids are formed by the WGD, that is whole genome diploidization, in the recent hybrid, such that there are now 2 copies of each parental contribution to the genome. Okay. So here we are having this, and this from this particular diploid species, and this one, and this one, and this one from this particular species. So every genome is present as the parental, and we are having this 2 set of chromosome in each form. Though that is the allopolyploids. Okay, so this is all about this session. We will meet with one more session to understand the 2 other different polyploids. Thank you very much.